Hi everyone, welcome to your online lecture for the course Human Anatomy and Physiology 1. My name is Dr. Patricia Riley, and in today's lecture we're covering cell function or cell physiology. We'll look at the transporting of substances in and out of the cell metabolism, and the generation of energy. So how do substances travel, or how are they transported in and out of the cell? Well, remember that the cell membranes are made up of phospholipids, and these phospholipids work to control which substances can move in and out of the cell. And we call the cell membrane a semi-permeable membrane or a selectively permeable membrane because it's very specific on what it will let in and out and how. And this membrane is going to work to keep two bodies of water, that is outside of the cell and inside of the cell, from one another. So it's separating those environments. Two basic types of transport mechanisms that can permit the movement of substances into and out of the cell include passive transport and active transport. Let's start off with passive transport. And passive transport is where substances or water can move down their concentration gradient. So they're moving from an area where they're in higher concentration to an area of lower concentration. And so you can see that occurring in this image here, where this is inside the cell, this is outside the cell. These substances are in higher concentration inside the cell, and they can move passively to an area of lower concentration outside the cell. This does not require any energy. Again, it's passive, so passive means it does not require energy. Two examples include osmosis and diffusion. Osmosis refers to the movement specifically of water from an area of high concentration to an area of low concentration, whereas diffusion is the movement of solutes, so things that are dissolved, as well as water across the membrane. We'll elaborate on many of the details on this slide in upcoming slides, but right now an important distinction between these two is that osmosis does refer specifically to water. So now on to active transport. Active transport is the opposite of passive in that substances are moving against rather than down against their concentration gradient from an area where they're in low concentration to an area where they're in high concentration. And we can see that in this image here, where substances are in higher concentration inside the cell and lower outside the cell, but the movement is in the opposite direction than what we saw with passive transport, moving into the cell. And it requires, because it's moving against its concentration gradient, this requires energy and the existence of pumps. And we can see listed here that an example of active transport is the use of pumps that are in the cell membrane that can actively pump substances from where they're in lower concentration to higher concentration, but this does require energy. Okay, energy is required. Another example of how and why active transport exists is because there are times where we need to move substances in bulk into or out of the cell, and that's where vesicles are involved. And so you can see an example here where substances in bulk are being actively transported out of the cell. And in this case, substances are being actively transported in bulk into the cell, and you can see vesicles mediate that. And energy, of course, is required for that because we know it's an active transport mechanism. And the two examples of bulk movement are endocytosis and exocytosis. And those are shown in this diagram here. And we'll learn more about this later, but endocytosis is the movement of substances into a cell. So that's shown here, endocytosis bringing substances in. And exocytosis, think EX for exit, substances are in bulk going to be leaving the cell. And this also requires the use of energy. Let's elaborate now on the concept of passive movement. So passive transport. We're going to be looking specifically at passive movement of solutes and water. And I'd mentioned earlier that this was called diffusion. So diffusion is a passive process. Obviously, we've already said that. And this is where small particles will have this tendency to want to diffuse or spread out in a given space. And these molecules have the ability to just diffuse through or spread through the membranes, so the cell membrane. And we know that in the case of diffusion, a passive movement, that we're going to be moving from an area of higher concentration to an area of lower concentration, so down a concentration gradient. And we know that this does not require any energy expenditure. So let's look at an example, just a basic example of a beaker of water and what happens when a lump of sugar is placed in it. So what we have here is a lump of sugar being placed in water, and what you can see happening over time is the sugar molecules, they started off very dense and packed, and then over time, these sugar molecules are going to collide and they're going to move from where they are in higher concentration and spread out from each other. Okay, or diffuse from one another to where there is a lower concentration of that substance. And so eventually there's going to be somewhat of an even distribution of those sugar molecules. In the second example, there is a barrier placed, which is representing a membrane, just like we have our plasma membrane that separates the inside and the outside of the cell. And the barrier here is designed to allow for the movement of both water and solutes through the membrane through a passive process called diffusion. And when we look at the first image, we have 10% solute and 20% solute on either side of the membrane. And then what's going to happen over time is it's going to naturally want to create a state of equilibrium. So you're going to get the movement of water and solutes. You can see here the solutes are moving from where they're in higher concentration to an area of lower concentration, trying to create equilibrium. And it's thought to reach equilibrium, this solution, when we have no net change in the amount of water and solutes on each side of the membrane. And you can see that represented by now an equal amount of solutes on either side of the membrane. So remember, this does not require any energy. And the larger arrows are representing more movement of that particular substance through diffusion. Now let's look closer at the specific passive transport processes we talked about and look at the difference between osmosis and diffusion. Osmosis, I had already told you, refers very specifically to water, and osmosis is considered to be, we know, a passive movement of water through a semi-permeable membrane. And we can see an example shown here where water is going to move from where it's in higher concentration to where it's in lower concentration. And this is osmosis because these substances here, these larger particles, it's a protein called albumin, is not able to penetrate through the barrier. And so it's going to be water that is going to passively move through osmosis from where it's in higher concentration to where it's in lower concentration. And it's sort of, if you can see the number here, 5% albumin to 10% albumin, and now we have 7.5 on this side. We've diluted this substance, or this solution, I should say. So then we can say that water, in this example of osmosis, water is what's moving across the membrane, and what's not moving are the solutes, in this case albumin. Equilibrium is reached when we have the same amount of those solutes present on both sides or in both solutions. And when there's a different volume of water on both sides, and you can see that there's less water here than there is here because water has moved from this side to this side in order to dilute that solution. Now, that's osmosis. Let's look at diffusion a little bit more, remind you about some of the things we've talked about already. So we've seen this diagram on the previous slide. Diffusion we know is going to involve the movement of both water and solutes compared to osmosis, which is just the movement of water. And equilibrium is going to be reached once there's the same concentration of solutes on both sides. So here's our equilibrium, same concentration of solutes on both sides, but both solutes and water are moving in order to achieve this, whereas with osmosis, it's just water that's achieving this. And so we also then see the same volume of water on both sides compared to what we saw with osmosis. We're going to have a look now at some examples of the different types of passive transport that exist within the body. And this does not require energy, so it's just the passive movement of substances. You can see there are a lot of different examples listed here. And these are all types of passive transport, but there are differences in how substances are moved passively into or out of the cell. You can see the word diffusion here a lot, and we've talked a lot about diffusion already, but there are different types of diffusion, with the most simple being simple diffusion, where molecules can just easily and simply move through the membrane. It doesn't need any special channels or carriers in order to get through the cell. It can just go right through the membrane. And there are only going to be specific substances that have the ability to just diffuse right through the membrane. Now, facilitated diffusion, on the other hand, facilitate means to help. This is where, in order for diffusion to occur of certain substances, they need to be helped or facilitated. And so this requires the use of pores or channels. Facilitated diffusion is going to allow for very selective passive transport of water, and it's also going to be allowing for the movement of high volumes, very fast movement of water when it's needed. We have some examples here listed below of facilitated diffusion. So channel-mediated, carrier-mediated, gated ion, and aquaporins then are examples of facilitated diffusion. So let's look at each one. The first one being channel-mediated diffusion. A couple of important points about channel-mediated diffusion is that, one, it means there's going to be some sort of channel that's present, an opening that can allow for the substance to move through. And we can see that quite simply here. Here's an integral membrane protein that is embedded in the plasma membrane that's going to allow for the movement of substances that can't otherwise get in through this particular channel. Now, it's important to know that these channels are very specific in that it's designed for the movement of a specific substance. These channels can be opened by being stimulated. They can be closed. They can also be inactive. It's going to vary depending on what's needed by the cell. Another example here of channel-mediated diffusion, you can see this channel that's present here within the plasma membrane that's allowing, when it's open, allowing specific substances in, and then when it's closed, preventing those substances of any kind from entering into the cell. Our next example is carrier-mediated diffusion. Carrier-mediated diffusion is going to use another type of membrane protein which actually carries or grabs the material and brings it into the cell. It's going to be very specific In other words, this integral membrane protein is meant specifically for this molecule, and so it will grab onto it rather than it just moving through the channel like this example. It's carrying it into the cell, and you can see it's being brought in and then it's being pushed into the cell. Next up, we have gated ion channels, and so these are just channels that are almost controlled by a gate then that can be opened or closed. And so we can call this example here, where we spoke about being channel mediated diffusion, that it can also be called a gated ion channel that controls the movement of certain ions in and out of the cell. And we're going to come back to that a bit later. Then the last type of facilitated diffusion is an aquaporin. An aquaporin is very selectively going to be moving water, but it can be used to move a lot of water. So aqua means water, porin is referring to pores. So it's water moving through pores that are present in the cell membrane. Because normally we couldn't get a lot of water in the cell unless there was a pore that would allow it to enter the cell, because we know that water doesn't pass through a lipid membrane very easily. And so this here is an example of an aquaporin that's present that allows specifically the movement of water through it. And then finally, we have osmosis. We've talked about osmosis being the selective passive transport specifically of water. So this is just in normal amounts, not large, fast amounts. And if we want a lot of water to move into the cell, we would need the presence of aquaporins. But osmosis is a method of getting water in and out of the cell. Let's apply osmosis now to the human body, to human cells. And we're going to be looking at two terms, osmosis, which we've already defined, and tonicity. We're going to define osmotic pressure, though. Osmotic pressure would be water pressure that develops in a solution as a result of osmosis into a solution. We call this osmotic pressure. Tonicity refers to osmotic pressure outside of the cell relative to the inside of the cell. Or if we were looking on our previous slide, it would be the relative osmotic pressure of two fluids. So we had fluid on one side of the membrane and fluid on the other side of the membrane. But we're going to apply it to human cells, but specifically we're going to look at red blood cells. Now, red blood cells are going to be found within our blood vessels, and they're bathing or traveling in a solution that we call plasma. And we need to have the perfect environment for our red blood cells to maintain the shape and function. And so what we're going to look at is three situations for a red blood cell. Red blood cells being in an isotonic solution, in a hypertonic solution, or in a hypotonic solution. And then we're going to see what would happen to that red blood cell in that environment. And this will show you the importance of osmosis and tonicity homeostasis in our body. So here at the top, we have red blood cells, and they have water inside them. And there's going to be a specific osmotic pressure within the red blood cell and a specific tonicity within the red blood cell. And so we're going to see what happens when we drop these red blood cells in these three different solutions. Another thing I wanted to mention before we look at these examples, when we hear the word tonicity, I want you to think of solutes as well. And the greater amount of solutes present in a solution, the greater the tone or the tonicity. So let's first look at isotonic solutions. Iso means the same. Tonic refers to tone, and this has to do with the solutes. And an isotonic solution then is going to be one that has the same amount of tonicity inside the red blood cell as the tonicity outside of the red blood cell, so in the solution. And so because of this, there's no net movement of water. So let's see what that looks like. This here's an example of a red blood cell in an isotonic solution. And so we can say that the tonicity outside the cell in the solution here is the same as the tonicity of the red blood cell. And water likes to follow tonicity or follow solutes. So because it's the same inside and outside, there's no net movement of water. We don't have large amounts of water flooding in the cell or large amounts of water flooding out of the cell. So this is the scenario that we want within our body. Cells are able to maintain their size and shape, and they're not going to be damaged in this solution. Now let's look at another example where we have a hypertonic solution. Hyper is going to refer to meaning that there's a lot of something, and tone we know means, tonic means tone or tonicity. And so this means there's going to be a lot of or high amounts of tone or tonicity. So in this particular solution that's hypertonic, it means there's higher amounts of things dissolved in it, so solutes, in the solution compared to what's inside the cell. And water likes to follow tone or follow, we can say, solutes. And so water is going to move from the inside of the cell to the outside of the cell, following where the higher tone is present in order to sort of dilute the area. That's its desire, is to move to an area of higher tonicity. So here's showing you that water flows out of the cell. We can see this is the example here where water is leaving the cell, moving to an area where it can sort of dilute the solution. And if water is leaving the red blood cell, it's causing the red blood cell to shrivel or shrink in size. And this is called crenation. So cell shrinking is called crenation, and this can have some serious side effects on the body if it's not corrected. In other words, if the solution that the red blood cells are bathed in is not corrected soon, then it can have long-lasting damage and, well, can cause death. You can think of the red blood cell in this hypertonic solution as becoming dehydrated. It's losing a lot of its water and therefore changing its shape. In our final example of a hypotonic solution, hypo means low and tonic, of course, is tone or tonicity, relating to the amount of solutes that are present in it. And so this is a solution that has lower amounts of solutes, so things dissolved in it, than inside the cell. More solutes is going to increase tonicity in that area. So water likes to follow solutes or follow tone. So water is going to move into the cell. It's going to move from where it's in higher concentration to where it's in lower concentration. And this is going to cause the cell to swell. So you can see water rushing into the cell, and too much water can cause it to increase in size. So it's moving in to dilute now the cell because there's a lot more tone in the cell than outside of the cell. So as I said, this can cause the cell to swell or become bigger. And if it isn't corrected, it can cause cell lysis, which is actually rupturing or damage to the cell walls. And if that happens, it can't be corrected. So this can also be an important concept from a medical perspective when people are given fluids into their body. You have to be careful because you could cause cell crenation or cell lysis if the proper solution for that situation is not administered. So in all of these cases, what's happening is the solutes that are present in the solution or in the red blood cell, they're staying where they are. And so the only way to balance things out and dilute areas is for water to move. And water, via osmosis, is going to want to follow where there are more solutes present, where there is greater tone. Okay, looking at the top of the slide now so you know what we're getting into, we're going to elaborate on active transport, which was introduced earlier in the lecture. We know that active transport of substances across a membrane requires energy to do work, so I want you to associate the term active with needing energy, and we know that that energy is in the form of ATP, and it requires energy because it's moving substances really against their concentration gradient. It's like you're trying to push through resistance, and so you need energy to do that. And we talked about pushing a ball up a hill requires energy, and that's a very active process, but just tapping a ball to go down a hill does not require energy, and that is passive. So what we're going to talk about here is a little bit of elaboration on these images before we go into the different types of active transport. But we have really two purposes when it comes to active transport. We're either trying to return a substance back to their original location, so pushing it back to where it was, or we're trying to move substances in bulk. And to move something in bulk that might be, say, heavier, let's say, to help you understand it, that's going to require energy. In this image here, we have substances moving from low to high pressure, so it's like they're having to push through to resistance in order to get back to where they were. We also have an example here where substances are being exported in bulk out of the cell or being brought into the cell, and you'll see soon that that also requires energy. And then in this third example, we have this active transport pump that actively using energy is going to pump substances out and into the cell. So let's get into a bit more detail. All right, so the types of active transport that require energy include pumps, also called active transport pumps, and vesicle-mediated active transport. So the first example is the pump, which acts to move or pump substances against their concentration gradient. So it's moving in the opposite direction, really working to concentrate substances more on one side of the membrane, versus, say, diffusion, which was trying to balance things out on either side. So here we have an example of a pump where we have this integral membrane protein that's pumping solutes from the inside of the cell where they're in lower concentration against their concentration gradient to an area where there's more solutes present, and you can see that ATP is required. So vesicle-mediated active pumps, these are really like containers, like the vesicles are like containers that move substances, and substances can leave or they can come in via a vesicle, and we saw that on a previous image, but we'll look at two different types of vesicle-mediated transport. And they are here. So these two types of vesicle-mediated transport, exocytosis and endocytosis, were introduced already to you, but to look a little bit more closely at them, the first one is exocytosis. Exocytosis, EX, think of exit, allows to take new material that's been made in the cell. Let's say we make a protein, and that protein is meant to be part of the plasma membrane, let's say as an integral membrane protein. We can carry it from where it's produced, we know at the ribosome, and then it's going to make its way through the Golgi apparatus, and then it's going to be packaged in a vesicle and brought to the plasma membrane where it's going to be integrated into the plasma membrane. So it's sort of being exported, but it remains in the plasma membrane. Then we have other instances where we produce something in the cell that is meant to leave the cell. So for example, we could produce a hormone in the cell, and many hormones are proteins, and then we need to get it out of the cell, and it needs to get into the blood. So what it needs to do is leave the cell, and it's going to leave through a vesicle, like a container. So here's a simple diagram showing you that we've made this substance in the cell, but what we need to do is get it out of the cell. And so it travels to the cell wall in a vesicle or a container, and then it sort of binds with the plasma membrane wall and then is exported or able to leave the cell. The other example is just the opposite, and that's endocytosis. This is entering. Think of EN is entering. Substances need to enter the cell for a variety of different reasons. So for example, we could have maybe there's something in the plasma membrane like an old integral membrane protein that needs to be broken down and digested. It can be packaged up sort of into a vesicle that pinches off the plasma membrane wall and brought into the cell so that we can break it down. Another example would be something that's extracellular, so outside the cell, is going to be brought into the cell, and it could be something that the cell actually needs to use, but it could also be something foreign that's being brought into the cell in hopes that our cell will then dispose of it. For example, we talked about lysosomes would bind with it and destroy any foreign material. And so here's our example here where some substance we want to bring into the cell gets into the cell by binding to the plasma membrane, and then a vesicle starts to sort of pinch off, and here's that container vesicle in the cell. We're going to talk now about two common active transport pumps that exist in the body. And the first one that we're going to look at is called the calcium pump, and we're going to look at the purpose of this pump, really just introducing it. It will come up again when we look at the muscle unit, because these pumps are found in all muscle types, and calcium is something that's required for proper muscle function, but it needs to still be controlled. So what will happen when we want a muscle contraction to occur in our body is we're going to have calcium ions that diffuse into the intracellular, into the cell, and they do so through these gated ion channels that we talked about earlier. So these are channels that are gated, but they will permit, under certain circumstances, the movement of ions through it. So after muscle contraction has occurred, we need to get rid of this excess calcium. One, because we want the muscles to be able to relax, but two, because we don't want calcium to just build up in the cell. So in order for relaxation to occur of the muscle, we need to get calcium back to its original location, so wherever it originally was. Sometimes the calcium comes from outside the cell, so extracellular fluid. In other cases, it's stored within organelles. And you might recall from class talking about how the smooth endoplasmic reticulum is an organelle in our cells that actually stores calcium. And so a calcium pump then is needed for this purpose, to get rid of that calcium once we no longer need it, returning it back to its original location. And that has to be done actively, and so it requires energy. So what we can see in this image here in terms of how this calcium pump works is you're going to have calcium that enters into this pump that's specific for it, and it's going to be actively pumped then out of the cell or wherever it is that it's going to be stored. And we know that ATP is being used for that because it's active. Now, ATP is just a form of stored energy. We've talked about how ATP needs to break down into ADP in order to release its energy. And so when this happens, you can see the ADP here plus an inorganic phosphate, and it's going to allow for the actual movement of these calcium ions then in this example, out of the cell and back into the extracellular fluid where it was to begin with. So you don't need to worry about all the details that I'm not speaking of on here. We will be talking about this in more detail and making sense of it once we get to the muscle unit. So that was the calcium pump. Another common active transport pump is the sodium-potassium pump, or the NAK+ ATPase pump. And this, just like with calcium, is designed to pump solutes to where they originally were or against their concentration gradient. So we're looking specifically at potassium and sodium. And this type of pump is found in most cells in the body. So I'm going to give you a reminder from a previous lecture, and I know this is a bit small, but what I want to remind you of is that extracellularly, so outside of the cell, we should normally have high concentrations of sodium. And this is just breaking it up into your interstitial fluid, which is in between cells, and your blood plasma. But what I want you to know is extracellular fluid outside the cell, we have more sodium. But inside the cell, so intracellular fluid, specifically in the cytosol, which is the watery part of the cell, we have higher levels of potassium. So make note of that, because that's important for you to understand that normally we should have more potassium in the cell and more sodium outside of the cell. And how that's maintained is through a process using the sodium-potassium pump. So let's look at these images here. This was an example that I showed you earlier where we have this sodium-potassium pump, and then it's showing you here that 2 potassium are being pumped in the cell and 3 sodium are being pumped out of the cell, and specifically at that ratio. And this helps maintain the normal levels of sodium and potassium inside versus outside of the cell, going back to what it should be. But if we look at this image here, what we can see is that we have this enzyme that's an integral membrane protein that's embedded within the plasma membrane wall. And it's specifically called a sodium-potassium ATPase integral membrane protein. And what you can see happening is 3 sodium are going to bind here, and energy is required. And at the same time as this sodium is being pumped out of the cell, we have 2 potassium coming into the cell. And you can see that the pump is actually changing its shape, or the enzyme is changing its shape in order to do its job when you compare the different pictures as we progress. So this is restoring then the normal balance in the cell, and it allows for the maintenance of that lower amount of sodium that should be present in the cell compared to outside the cell, and the higher amounts of potassium in the cell compared to outside the cell. So what we just did is we spoke about active transport, specifically looking at pumps, and the pumps that we looked at were the sodium potassium pump and the calcium pump. Now what we're going to do is look at that other type of active transport system that we spoke briefly about, and that's called vesicles. And we talked about exocytosis and endocytosis, but now we get to go into just a little bit more detail. Okay, the first thing we're going to look at is exocytosis. We know that exocytosis, that EX, is referring to the exiting of substances or leaving, either going to the cell wall to be integrated into the cell wall or leaving the cell. We call that exocytosis. So what we have here are larger compounds that are going to be, say, manufactured in the cell, are going to be put into a vesicle or a container, and they're going to reach the cell membrane. And one of the ways that this happens is through the use of the cytoskeleton. And remember, when we talked about the cytoskeleton, we said that one of the features of the cytoskeleton was to allow for the movement of certain substances, including vesicles, within the cell. And so the cytoskeleton then is helping that vesicle move towards the plasma membrane. And at that point, it will either be exiting and leaving the cell and becoming part of the extracellular fluid, or it's going to be embedded into the plasma membrane, depending on what it is. So that's shown here. This is showing you actually just exiting. So this could be a hormone that's leaving the cell and destined to end up in the blood. Okay, now we have these two here. We spoke about endocytosis, but we have two types of endocytosis. Remember, endocytosis, N is referring to entering the cell, but there are two different types. Phagocytosis, and you will hear this term multiple times throughout your education, and pinocytosis, and you won't hear this quite as much, but both are still important for you to know right now. So phagocytosis is cell eating, and pinocytosis is referred to as cell drinking, and that's just referring to the types of substances that are entering the cell. So with phagocytosis, this is where there are larger particles, okay, so cell eating, okay, larger particles that are outside the cell and then brought into the cell. So they're engulfed by the plasma membrane to form that vesicle that we spoke about. So what's happening here, and I'll just switch to a pen, is we have a particle, a large particle, that is engulfed. You can see it sort of looks like the cell is trying to eat it, engulfed by the plasma membrane. Then it pinches off and forms this vesicle. Now, let's say this was something that was foreign or something bad that we wanted to break up. Then it would fuse with a lysosome, and you can see that that fusion is happening here. And then that lysosome is going to mix its digestive enzymes and break down whatever substance that is. So that's an example of endocytosis. It's not always something bad that we're bringing into the cell, but when we talk about phagocytosis, we're bringing something into the cell eating, and then doing something with it once it's inside the cell. Pinocytosis is referred to as cell drinking because it's fluid that's being brought into the cell rather than large particles. There will be some dissolved substances as well within that fluid, and the same process occurs where it's brought in and engulfed by the plasma membrane to form a vesicle. The difference here is it would be a fluid with dissolved substances in it compared to a large particle. Now, these events are not random. In most cases, what our body should be doing is identifying what it's bringing into the cell. We should identify, oh, is this a bad thing? If it is, let's destroy it. Oh, is this a good thing? We shouldn't destroy it. But our body is not always able to necessarily identify what it's bringing into the body, which is why our cells can be harmed with many different conditions. So just like if somebody was to say knock on your door, and the door would be the plasma membrane, you would want to identify whether or not you know that person and you want to let them in. And so what we're going to talk about next is something called receptor-mediated endocytosis. So this is a way to identify what the cell is bringing in as something that we want to bring in. All right, so here we're still, of course, on active transport, receptor-mediated endocytosis, so substances entering the cell. Remember back to our conversation about integral membrane proteins when we talked about cell structure. I had mentioned that some of the integral membrane proteins are actually receptors, and they're these receptors that are just built into the plasma membrane, and they're able to recognize what comes up to it. And what it will do is have we have so many different receptors present on our cells that are meant to identify certain substances. And it would then be a perfect lock and key fit in order for that receptor to permit that substance to enter the cell. If it is something that we want to bring in the cell, then the process of endocytosis occurs, and it's the cytoskeleton that actually contributes to the movement of that plasma membrane inward to form that vesicle or container or pocket to bring the contents in the cell. So this is a bit of a review from what we've been speaking about, but let's go through this step by step. So we have these receptors on the cell membrane that are specifically meant to bind with this substance. Now let's say this is a bacteria that binds with the cell. The cell will recognize it as being foreign, and the cytoskeleton will allow for it to pull inward to form that vesicle and bring it then in the cell. And then if it was something bad like a bacteria, then we would have a lysosome that would fuse with this vesicle and spit its contents inside, which are the digestive enzymes, to break it down and kill it. And this is one of the many methods that our body has to keep us healthy. And so the final point to make here is that our cells contain all of these different receptors that are integral membrane proteins that are integrated into our cell walls, our plasma membrane, to identify all of this different substance that come into contact with it. And these receptors are going to be very specific to certain substances. Now we're moving on to a different topic called cell metabolism, and we're going to look at the role of enzymes and what they do within body cells. We've seen this slide already, but I'm going to remind you of some of these chemical reactions. So we have anabolism, which we know an example, well, let's define it first. This is where we're going to be building larger molecules from smaller ones. So what we have are these two smaller molecules, and we're going to form a polymer, a bigger one. And we also know that this process usually uses energy in order to build something or make something. And an example is a dehydration synthesis reaction. So in this case, we're removing water in order to form a polymer, so something larger. Okay, building a larger molecule. Whereas with catabolism, we are doing the opposite. We're breaking these larger molecules into smaller ones. So here's the opposite. Our larger polymer is being broken down into smaller ones, smaller molecules. And an example of this is a hydrolysis reaction. So the addition of water to this polymer will break the bond, forming two smaller molecules. And this one normally produces energy rather than utilizing energy. So in order for all of this to happen, we need something that drives the reaction. And the driving force are going to be enzymes, which is our next topic today. And enzymes are proteins that have a particular function. They're going to function as a catalyst. And a catalyst is what drives the reaction. So cell metabolism is just a set of chemical reactions that are going to be occurring within the cell. And enzymes play a big role in cell metabolism because they catalyze the reaction. And why this is beneficial, and we'll see this shortly, why it's beneficial to use an enzyme is because it reduces the activation energy, or the amount of energy that we would need for that reaction to get started. So it really makes things more efficient. And so without enzymes, we would need a lot more energy to do the same thing. So let's see what that looks like. Let's see how enzymes play a role with metabolism. So just a quick overview, enzymes are going to be involved in regulating cell functions by having an influence on metabolic pathways. They allow for reactions to occur with less energy, using less energy to do it. When we look at an enzyme name, we can break it up to know what it is and what it does. The first part of the name of an enzyme indicates the substrate or the type of reaction that's going to occur, and I'll give you an example soon. And then the end part is almost always ACE. And so when you see ACE, it means enzyme. So in this example here, lactase, we have ACE representing an enzyme, and the lact part refers to the disaccharide lactose. So lactase is going to be breaking down lactose, which is a disaccharide, into its monosaccharide. DNA polymerase, well, we have DNA polymer, and then we have ACE as the enzyme. So this is going to be the enzyme that helps with the production of making DNA polymers. So enzymes then have specific names because they have very specific functions. Okay, very specific. Some of their properties, enzymes have the ability to allow for chemical reactions to be driven in both directions. So enzymes can be used in both directions. And enzymes are continually being destroyed and continually being replaced. So breaking down old ones and making new ones. And then enzymes are sometimes synthesized as inactive proenzymes. So what this means is we can have enzymes that are inactive proenzymes that are circulating around in our blood, and they become active when something else activates it. So it's like they're hanging around, these inactive proenzymes, waiting for something else to activate these enzymes or put it into active mode when it's needed. So let's look at some of these images here. First of all, here is DNA polymerase. That's the enzyme in cells that allows for the formation of the DNA polymer. This image here is meant to show you that enzymes can be used for both directions. So the formation of a polymer, but also the breaking of a polymer. And then this image here, this graph, is showing you that if we want to take reactant or substrate AB and form A and B, this chemical reaction is much more efficient and requires less energy if we use an enzyme. So here we have energy of the reaction with an enzyme, and you can see how much more when we don't have an enzyme. So that goes back to what I mentioned before, that enzymes lower the activation energy that's required in order to initiate a chemical reaction. So let's look at the chemical structure of enzymes and what they're made of. We know that they're functional proteins, but they take on this complex shape that allows them to participate and catalyze a chemical reaction. Let's look at the components of an enzymatic reaction. So the three main components include the substrate. The substrate is the raw material before the chemical reaction. So it's this substance here, that molecule AB, that is the substrate. So it's in its raw form. The product, as you would expect, is going to be what you end up with. So after the enzyme acts on this substrate, AB, we're going to end up with product A and B. It's broken down, AB, into A and B. And then the active site is going to be the part of the enzyme that fits the substrate. So the active site here, this structure of this enzyme, is designed specifically for the function of having this particular molecule bind to it. So it's like a lock and key fit. Okay, so we have the substrate that binds to the active site on the enzyme, and we have the formation of molecule A and molecule B as the product. But let's look at that a little bit closer. So first thing that we have happening is substrate AB is going to fit into the active site of the enzyme like a lock and key. The next thing that's going to happen is the enzyme is going to change its conformation or its shape, which is going to be where it's becoming activated. So it surrounds the substrate of interest. The enzyme is going to allow for a catabolic reaction of the substrate. Why catabolic? Well, we're taking AB and breaking it to A and B, so we know it's breaking down. So now we form molecules A and B, which are still attached to the enzyme in this image. And then the enzyme is going to resume its original shape, which allows for molecules A and molecules B to be let go and released. and it maintains then the enzyme returns, I should say, to its native shape. And the enzyme is now ready again to act as a catalyst for another catabolic reaction of another AB molecule. So I've already said it, but what type of reaction is this? It's a catabolic reaction. We have many enzymes in our body, many enzymes. But what I want to talk about now is that we actually need, in some cases, many to work together to achieve a result. And so in this image here, you can see that the initial substrate that binds to the enzyme will then produce intermediate A, which is then required to bind to the second enzyme to produce intermediate B, which will have to bind to enzyme 3 to produce intermediate C, which will bind to enzyme 4 in order to produce the end product. So in this case, we can say that the product of one enzyme ends up really being the substrate for the next enzyme. So when we produce intermediate B as a product, it becomes the substrate then for this enzyme to act on, which will form intermediate C. And so in this case, we need all 4 enzymes to be working to form this end product. And if one is not working, then we will not be able to form the end product. And so in this example, the enzymes are embedded in the cell membrane, but enzymes can also be found within the cytosol or in other areas, but we're just showing you what happens in the cell wall right now. And letting you know that, one, you can have many enzymes required just to produce one end product, and that you can turn off that entire metabolic pathway by one enzyme not working. So this is a summary of what I just spoke about, and I'll bring up the answers, but you can go through this on your own. And it's putting it all together from what I spoke about prior. So you can take a screenshot of this if you want to enter, or pause the video and fill this in, or fill it out yourself and check it later just to check your understanding. Okay, we're almost through. Enzymes can sometimes be influenced by what's called an allosteric effector, something that is physical or chemical that can affect the enzyme. So it can activate an enzyme, it can inhibit an enzyme, but ultimately what this allosteric effector does is it's going to be changing the shape of the enzyme molecule, which changes its active site. And we'll see what that looks like. But some examples of things that are considered allosteric effectors that again can change the shape of the enzyme is temperature, pH, ionizing radiation, cofactors, end products of certain metabolic pathways. Many different variables can influence how an enzyme functions. So let's look at what would happen if we had an allosteric effector that inhibited an enzyme, what it would do to it. So here's an enzyme. Here's what is considered the active site on the enzyme. So there would be a very specific structure that would fit in this active site. An allosteric effector that inhibits the effect of an enzyme will bind to the enzyme and, as you can see, change its shape. So it makes the active site now distorted. So now that enzyme is not able to function as it should. We can also have allosteric effectors that actually activate an enzyme. So in this case, it's binding to this enzyme that is currently inactive because the active site has been distorted, and it corrects for that and makes it functional again. So it's like it changes the lock so that the normal key cannot fit into it when it's inhibiting it. We're going to spend a moment talking about some of these allosteric effectors and the influence that they can have, temperature and pH to start with. And what's important for you to know is that temperature of the body, the core temperature, has to always be within a very narrow range in order for enzymes to function properly. And so if we go outside that normal range, here we have optimal. If we're outside of that, then it changes how enzymes can function. And on your slides, you can just copy the text here and put it into your notes, and then you can just click on this and delete it to show what's underneath. pH is the same thing. In order for certain enzymes to work in our body, and here are a couple examples, pepsin and trypsin, and these are involved in digestion, they can only operate under within a specific range of pH, otherwise they cannot operate. Now let's look at what happens if there is no allosteric effector. Without inhibition, then we would continue to produce the end product. So we need regulation. And in some cases, when there isn't an allosteric effector, instead it's the end product that becomes the allosteric effector. So what that means is that, let's look in this example here. If we have a substrate and we have all these enzymes working on it and we produce an end product, if we don't have any inhibition, then the end product continues to produce. So what happens is we have this feedback where the end product, and it's negative feedback, a negative feedback loop. We produce our product, and then the product is sort of fed back so that we realize we're producing this product, we don't need any more, and this will stop the process. So the end product ends up being the allosteric effector. And that was listed on one of the previous slides about one of our types of allosteric effectors is the end product. On our final topic, we're going to talk about catabolism and cellular respiration. There are 3 different types of cellular respiration we have here: glycolysis, citric acid cycle, and the electron transport chain. I want you to think of cellular respiration as sort of being like cell breathing that ultimately produces the energy that we need for survival. So what happens with cellular respiration is our cells are going to take glucose from the food that we eat and produce energy. So let's see for a moment what that looks like. All 3 processes of cellular respiration ultimately are going to produce ATP. Now I've pre-populated the numbers here, but try and follow along. So all 3 have the goal of producing ATP, which is the energy that we know we need to perform everything in our body. So let's look at each one individually. Glycolysis. Glycolysis occurs in the cytosol of the cell. So if we look up here, we have glucose, which is the energy that we need the nutrient that we need in order to produce ATP. So glucose will enter the cell and undergo glycolysis. And glycolysis is going to occur in the cytoplasm or the cytosol of the cell, and it's going to produce ATP and NADH. So ATP and NADH, which we'll talk about shortly. What I need you to know is that glycolysis, it's this very complex sequential process, a step-by-step process that's regulated by many different enzymes, and you don't need to know it all, but it's summarized here for you to see the complexity of it. But glycolysis produces very little ATP compared to the other processes. And the reason for that is because the citric acid cycle and the electron transport system occur within the mitochondria. And you would remember that the mitochondria is the power plant of the cell where most energy is produced. So what will happen is when glucose goes through metabolic pathways in the cytosol, ultimately once we end up in the mitochondria, the citric acid cycle is going to occur, which is also a complex sequential process with each step-by-step being regulated by very specific enzymes. it's going to produce a whole lot more of ATP than glycolysis. It's also going to produce NADH and FADH. Then the electron transport system also occurring within the mitochondria. This is going to also produce ATP, but it's going to use energized electrons that were produced by glycolysis in the citric acid cycle. So we have the NADH and the FADH in order to produce a whole heck of a lot of ATP. So you only need to concern yourself with what I have spoken about in this lecture because there is a lot of detail in the diagrams that you do not need to be aware of. That's the end of this lecture. Thanks for listening.